Mine didn't.
It gives me great pleasure to introduce Dr. Eli Adashi, our graduate speaker. Eli did his medical education at Tel Aviv University Medical School in Israel, and then a residency at Tufts University School of Medicine. And then he did a REI fellowship. And in fact, I should have asked him about this. First year was at Hopkins with Howard Jones before he retired. And they moved out to the West Coast to San Diego, and he did 3 years there with Sam Yen and Warren Shui. And then worked for a number of years as director of REI at University of Maryland. And then from there, moved out way out to Utah, Salt Lake City, where he was chairman of OB/GYN at the University of Utah. And then from there, moved on to become the dean of the School of Medicine at the Warren Alpert School of Medicine in Brown University. And now he's retired from that position, but is professor emeritus at Brown and very, very active in terms of publishing literature, and very proliferative in terms of into various intellectual aspects of and controversial aspects of our times in OB/GYN. He's also, I should say, the past president of the Society of Reproductive Endocrinology, ASRM, and past president of the SGI, Society of Gynecological Investigation, as an elected member of the National Academy of Medicine. So it's really an honor and a privilege to have him give us a lecture today. And it's a especially interesting topic I think we're all going to find unusual. And I welcome Dr. Adashi to the Zoom podium. Thanks so much, Eli, for being here today. We really appreciate it.
Thank you, Richard. And I like the term Zoom podium, although the last time I visited your university was in person. And in fact, we've done that probably several times in the past. In some respects, I miss that. I appreciate the power of Zoom, but I recently wrote a paper, which you may or may not have seen, arguing in favor of the in-person grand rounds. But best I can tell, it's a losing battle. And it's increasingly the rare example of grand rounds, which more and more seems to be Zoom dependent. I chose today's topic out of interest. It's a topic that is clearly futuristic in nature. It may or may not happen in my lifetime, but the technology is clearly coming along, and definitely in the lifetime of the young members of the audience, I suspect this may well become a reality. And so it struck me as of interest to explore at this time, even though it clearly means reimagining the whole notion of reproduction. What I will try to explore with you today is, first of all, the number of same-sex US households, the fertility realities today of same-sex couples, and the state of so-called state infertility mandates as they apply to same-sex couples. So let's begin with the first. And to do that, I turn to the United States Census Bureau, which interestingly keeps track of the total number of same-sex households in the United States, and seems to have done so since 2008. I cannot speak to the methodology that's involved. I also cannot speak to the accuracy thereof. But to the degree that we want to take the data as is, as shown here, and this is, of course, published information, it would appear that by 2022, there were approximately 1.3 million same-sex households in the United States. About 700,000 of those were married couples, and approximately 500,000 were unmarried same-sex couples. So this is the best one can really come up with in terms of the economy, if you will, of same-sex households in the United States. Now, if a same-sex couple wishes to have a child that is not entirely genetically their own, but if they wish to parent a child, there are relatively standard procedures in place today that can be offered. So for the same-sex female couple that is seeking conception, really the only missing requirement is the sperm donor. And once that is provided, then a pregnancy can ensue in one of the members of the couple. And that, in a sense, concludes the goal of that particular couple. The situation is more complicated, of course, in the case of same-sex male couples who wish to serve as parents of a child. In this case, in addition, they will require an egg donor because obviously none of the members of the couple can provide an egg. And that egg donor will have to be fertilized by a member of the same-sex male couple. And the resultant embryo, of course, would have to be carried by a female surrogate. So for same-sex male couples, it's a relatively involved operation, but it's doable and definitely has been accomplished. With respect to the current status of state infertility mandates, we can probably say the following. The first state to address this issue, although not right away, was the state of Maryland. By way of history, Maryland constituted the first state in the Union to legislate an infertility mandate, which at that point was directed not at same-sex couples, but rather at the conventional opposite sex couples. We wrote this up in the Journal of Fertility and Sterility in 1928. This is a photograph of the signing of the bill by the governor, who at the time was Governor Harry Rowe Hughes, a Democrat who supported the effort. But if we ask the question, did the 1985 Maryland infertility mandate, which was clearly unique and leading the way at the time, did it apply to same-sex couples? And the answer is no. In a word, same-sex couples were not being considered at the time, which requires fast forward 30 years or so to 2015, when the state of Maryland revisited the issue this time with same-sex infertility in mind. We wrote a short commentary in JAMA in 2015 on this very issue because we got wind of the possibility that a health insurance mandate for same-sex infertility was in the cards, which at the time, of course, was unheard of. As was mentioned by Dr. Brunson, I spent 15 years at the University of Maryland, and so I had certain connections. I obviously had a certain affinity to the state, and so I've been following it perhaps with greater affinity than some others may have. Bottom line is that during the tenure of Governor Lawrence J. Hogan Jr., a Republican, a state mandate for male couple infertility was finally passed. Now, Governor Hogan, who was a conservative Republican, would not sign the bill. But it so happens that the Maryland Constitution is home to something known as Article 2, Executive Department, Section 17, which allows in select cases the legislature to, in a sense, sign off on a bill and render it active law, independent of the governor's signature. And that is what happened here. The essence of the legislation read, prohibiting insurers, nonprofit health service plans, and health maintenance organizations from requiring specified conditions of coverage for specified infertility benefits for a patient who is married to an individual of the same sex. It is of note that once Maryland legislated a same-sex infertility mandate, other states gradually followed. Not quite as many states as currently support heterosexual conception, but several states, including New Jersey, New York, Illinois, Colorado, Maine, and very recently, just a week or two ago, California had a same-sex infertility mandate signed by Governor Newsom, who is shown here in the process of announcing the same. Well, now that we have acquainted ourselves with at least the legislative facet of this possibility, the question is, how do we do this? How does it work? Which takes us back to the future and to assisted same-sex conception. There are two technologies that are clearly not in play right now in the human, but are very much all but guaranteed to one day be applied in the human and make it possible to proceed with same-sex conception. One of those technologies, which is far more advanced than the other, is known as in vitro gametogenesis, or IVG, which in the rodent has now been fully characterized, but has yet to be fully characterized and or accomplished in the human. The other technology, which is certainly much further off and may never be applied because of the perceived risks in the modification of genomic imprinting, nevertheless is feasible and can give rise to same-sex conception, as has been shown to be the case in rodents. And we will briefly cover that possibility. More relevant, though, is the notion of IVG. Just so you don't consider this an unheard of esoteric option, I'm showing here a recent review that saw press in Science magazine, which tells you that this technology has now achieved a level of credibility that is acknowledged by leading entities such as Science magazine. The New England Journal of Medicine, not to be outdone, published a similar paper that it commissioned from Mary Herbert and Azim Surani from the United Kingdom. Perhaps more telling, or most telling, is the conduct of a workshop on IVG in 2023 under the auspices of the National Academy of Medicine, which I happened to have chaired and led, and which dedicated 2.5 days, and of course, a large number of speakers, to the notion of in vitro gametogenesis and the future thereof. This is one more depiction of the National Academy of Medicine announcement of this particular workshop, which gave rise to a book which can be retrieved free of charge from the National Academy of Medicine, should any of you wish to pursue this possibility. It's probably high time, though, that we try to answer the question, what is in vitro gametogenesis? At its essence, in vitro gametogenesis is about the transformation of a somatic cell into what is known as an induced pluripotent stem cell, or iPSC, which in theory could be guided into any cell fate, including a gametogenic fate. Now, to do that, you have to know the cellular cues that have to be applied, the sequence by which they are applied, the timing thereof, and realizing that some of this has to happen in the context of a niche or an environment. And by that we mean that oocytes are, of course, surrounded by granulosa and theca cells, which are viewed as the niche of the female germline, whereas on the male side, the sperm are surrounded by Sertoli cells that subserve a similar function. So the niche is essentially the somatic cells that encase the germ cell element, which in the case of the oocyte includes the granulosa and theca cells, and in the case of the sperm, include the Sertoli cells. Having us laid out those general premises, let us now turn to what's involved in murine in vitro gametogenesis, and let's start with the female germline. This feat has been accomplished in 2016 by Katsuhiko Hayashi from Kyushu University in Japan in a paper published on the pages of Nature. And in that paper, Professor Hayashi describes the reconstitution in vitro of the entire cycle of the mouse female germline. He started the process with embryonic stem cells or induced pluripotent stem cells, converted them to so-called primordial germ cell-like cells, combined them with some niche or gonadosomatic cells to yield primary oocytes, eventually M2 oocytes, which are shown on the left lower hand side, which upon fertilization gave rise to blastocysts, which upon transfer to female surrogates gave rise to pups. So in a sense, starting with an embryonic stem cell and ending up with real pups, Professor Hayashi reconstituted the entire cycle of the mouse female germline in vitro, at least initially. Some of the same was accomplished for the male germline, which we will discuss with lesser details. A feat that is the work of Mitinori Saito from Kyoto University, probably the father of this field, who published this paper in Cell Stem Cell in 2021. The title being In Vitro Reconstitution of the Whole Male Germ Cell Development from Mouse Pluripotent Stem Cells. So far, of course, we have limited ourselves to the rodent, but perhaps we can say something, although, as I said, we have not gone all the way, but we can at least describe the state of the art with human IVG. Here again, Professor Saito is the leading protagonist. And in this science paper, he started the human effort with human primordial germ cell-like cell. He combined them with xenogeneic ovarian somatic cells, and then embarked on in vitro gametogenesis, but could take the process only as far as oogonia. He could not achieve M2 oocytes, which is really what it takes for fertilization to be accomplished and for an offspring to be generated. On the male side, we are in a similar situation, whereby the sequence leading human male in vitro gametogenesis all the way has yet to be accomplished. What was reported was a partial effort on the pages of Nature Communication by Professor Kotaro Sasaki from the University of Pennsylvania. titled Reconstitution of Pro-Spermatogonial Specification in Vitro from Human-Induced Pluripotent Stem Cells. So as we survey the circumstance today, we are obliged to say that human IVG still has a ways to go. That it will happen, I don't think, is in doubt. When, of course, is the question. Now, it so turns out that a modification of in vitro gametogenesis, otherwise the conduit to conventional parenthood, can also serve under very unique circumstances, which I will describe, as a conduit to bipaternal parenthood, an effort that has now been reported in the peer-reviewed literature. Now, all of us, including yourself, will be well within our rights to ask, is this really an attainable goal? I think the answer is yes, although as we sit here today, we cannot make that claim with complete confidence in that we have yet to get there. But all the indications are that given what we know, and given the collective scientific power out there, this is, in my view, just a matter of time. And as we've seen so far, and that's been always true, then and now, it's the rodent model that is likely to lead the way. And there really has been no human work to date on same-sex conception using in vitro gametogenesis. Human application, as I state here, really remains futuristic at this time. But as we look into the future today, I have a strong sense of confidence that it can and will happen. It's just a matter of time. How much time, of course, is anybody's guess. But the paper that gives us confidence that this may happen is the one shown here. Again, contributed by Katsuhiko Hayashi and published on the pages of Nature last year. And the title speaks for itself. But what in effect transpired here is shown in schematic terms in the subsequent slide. What I show here is a small commentary that accompanied the Nature paper I just described and is titled Eggs from Male Stem Cells Using an Error-Prone Procedure. But in a sense, it is showing offspring of a male couple, albeit in the rodent, of course. So as you might imagine, the process had to begin with XY-induced pluripotent stem cells that have to be converted to somehow to XX-induced pluripotent stem cells. And that happens in part spontaneously in culture, but is assisted by the application of a drug called reversin to the mix. And so spontaneously or in an assisted fashion, some of the cultured XY-induced pluripotent stem cells derived from a male mouse lose their Y chromosome in culture and, in fact, duplicate and spontaneously convert to XX-induced pluripotent stem cells, which is really the essential substrate from which to work. Isolated XX-induced pluripotent stem cells were then converted using conventional, I say in quotes, conventional in vitro gametogenesis procedures to oocytes. So meaning M2 oocytes, which we know how to do in the rodent. And those oocytes were then fertilized, and the embryos were transferred to surrogate female mice to yield what today really seems all but unbelievable, bipaternal offspring. And those are shown here. And the title of the article in question is Mice with Two Dads, which clearly is mind-boggling and titillating in more ways than one. The subtitle is Scientists Create Eggs from Male Cells. And obviously, we have seen that theoretically accomplished in culture. And when that happens, you can fertilize those eggs and you end up with pups, the genetic parents of which were two males. Now, I mentioned at the beginning that apart and distinct from the method we just described, there is, in theory, another way of arriving at the same destination of same-sex parenting, and that is the modification of genomic imprinting. Although this is feasible, and as I'll show you, has been carried out in the rodent, I would be very hesitant to apply this in the human because this is a fairly intrusive intervention which involves deleting certain elements and genes that carry inherent risks that don't seem warranted by the goal in question, especially since we have other more reliable means, perhaps, even of accomplishing the same. But in the interest of completeness, let me perhaps illustrate briefly how same-sex parenting could be achieved in the rodent through the modification of genomic imprinting. This technology actually has a longer life, or has been around all the way since 2007, wherein the whole issue of how one develops, in this case by maternal mouse embryos, has been studied and addressed in vitro with a generation of bimaternal rodent embryos. This is another depiction of a paper that the same investigators reported on in 2016. The paper was titled Birth of Fertile Bimaternal Offspring Following Intracytoplasmic Injection of Parthenogenetic Haploid Embryonic Stem Cells, which is to say that parthenogenesis is part and parcel of this procedure, a process that, yet again, I think we would be careful, if not reluctant, to apply to the human. In a paper that I just illustrated briefly, the two authors report on the birth of fertile bimaternal offspring following intracytoplasmic injection of parthenogenetic haploid embryonic stem cells. Even more recently, in 2018, the same team published a paper in Cell Stem Cell on the generation of bimaternal and bipaternal mice, in this case from hypomethylated or demethylated haploid embryonic stem cells with imprinting. As I said now multiple times, while this is elegant and impressive in a way, one would have to be very thoughtful before subjecting people to this technology. And as you know, of course, in our business, the regulatory mandate is it has to be safe and effective. Not just effective, but safe and effective. And it has to pass muster with the FDA, which applies the regulatory test of safety and efficacy. So if I had to predict, I would say that imprinting, although workable, is unlikely to pass muster with the FDA. But as we're sitting here, we're obviously looking into the future and speculating, but that's at least how I would view it today. So with this, I will conclude this somewhat futuristic tour of duty. And I apologize if some of you were looking forward to contemporary applied technology. But I do think it is useful, helpful, and instructive to look into the future once in a while and to get a sense of the possible. Whether or not it is doable and possible, of course, only time will tell. But on the assumption that some members of the audience are on the relatively young side, I would predict that some development along these lines is inevitable, although it may take a decade, 2 or 3, nobody knows, before this is actually completely implemented and applied to the human. So with that, I think I'm going to close and let Dr. Bronson take over from here. So thank you very much for the opportunity. I wish I was with you in person as we used to do, but I guess Zoom has doomed us to long distance lecturing.
Well, thank you so much, Dr. Adashi. I'd say, well, very controversial, controversial because it's in some ways a decade is not that far away, or 15 years. And so IVG may become something that's a reality. Let me open up first to anyone who may have any comments or questions from the audience.
Richard, I have a question.
Okay, go ahead. And
Okay.
identify yourself to everyone now.
Okay. Yeah. Dan Koenigsberg. Hi, Ellie.
Hello, Dan. How are you?
Good. So I know this isn't your work, but quite often when I hear the kind of basic research that you're discussing, it sometimes gets to a point where I say to myself, say what? What did you just say? And that had to do with when you were talking about taking the males and having them create XX oocytes, and that somehow there is this process where you get down to the pluripotential stem cells, and somehow some of these cells not only lose their Y chromosome, but they pick up an extra X chromosome. And that's unlike any kind of cell biology I ever learned. So I don't know if you can speak to that, but that just seemed more like magic than like the science I was taught.
Well, first,
I have a related question.
yeah,
My
please.
thought when I heard that, Ellie, was, what about XA activation? Is there anything in this, since one of the chromosomes, X chromosome isn't activated, is that process perturbed in any way, or is that even studied yet too?
Yeah. Well, let me first say that I'm delighted to see that Dan is on the line. It's been decades probably since we last met, which is unforgivable, but also probably undeniable. As to the question, let me just say, first of all, the people involved, Katsuhiko Hayashi and Michinori Saito, are, in my view, definitely first-class scientists, but probably a step beyond. And while it's not for us to project or predict if science prizes are ever going to be awarded to this work, these two are likely to be the leading candidates. It was 2013 when I was in Japan for another reason, but I booked a flight to Kyoto so as to meet Professor Saito in person in his lab, at which time Katsuhiko Hayashi was essentially a postdoc who worked with him. And I spent the day in Kyoto, and by then I was already fascinated by the work. And of course, it's been 10 years since then that we organized the National Academy of Medicine workshop, during which time, of course, a lot of progress has transpired. But I can say from that vantage point, plus the fact that the papers in question have all been peer-reviewed in leading journals such as Nature and Science, that this is valid basic science that at this point gives out the promise of eventual application. In a lecture I give separately just on in vitro gametogenesis, which involves heterosexual couples, we can already see that this technology, 20, 30, who knows how long from now, will expand IVF, in some ways replace it. And so what we have done today and what I do when I speak about IVG is try to peer into the future with no guarantees, of course, and no certainties. I will say also, given the source of the work from Kyoto University, that Kyoto University has what is known as a stem cell institute, and they have a Nobel Prize winner in their midst who was awarded the Nobel Prize for developing induced pluripotent stem cells. So Kyoto is ideally suited, given the quality of the people, the institutional support, the Nobel Prize backdrop, to really make something like this happen, and they have. None of us can sign on the dotted line or make a bet, but I think it is probably appropriate to speculate, and it's also probably less than desirable to just push this aside as meaningless speculation at this time. I think we're peering into the future, but it's not here yet.
Other comments, questions? So I'm not sure this question's answerable, but if you were, let's say, someone in the FDA, what might be the criteria to say, yes, this is something that should move forward? It's reached the point of actual clinical application, and what evidence needs to be there. Maybe that's not answerable, I don't know.
Right. Well, I happen to be speaking on a regular basis with Dr. Peter Marks, MD, PhD, who would have made the decision had this been submitted today. But as a result of this relationship, he and I have on several occasions discussed this issue. Of course, Richard, there will have to be clinical trials carried out, and the results made available before anything of this kind can have any chance of being considered by the FDA. But as you know, the critical decision points of the FDA are safety and efficacy. If the Japanese or another group carries out the appropriate clinical studies, which is not going to be simple and is not going to happen tomorrow, and if they can demonstrate that the process is safe, as best one can tell, in terms of the outcome, then they will receive a fair hearing, I believe, from a committee of experts that is invariably assembled by Dr. Marks in keeping with the expertise required. So in this case, somebody like yourself might be called in. Although, as I said several times today, this is likely going to be entertained decades from now, would be my guess, if ever. So I think the FDA, as it's currently constructed, is well suited to deal with this, but it will take a mountain of scientific advancement and a mountain of clinical trials to assemble the kind of body of work that could be presented to the FDA to even agreed to assemble a committee to deliberate the issue. So any way we look at this, since this is a futuristic subject, it's going to be a while. But unless something changes in the workings of the FDA is well prepared to deal with anything of this sort.
See, Maria Fisher has a question. Maria, do you want to unmute and ask the question?
Hi. So just again, thinking in the future, does this suggest that someone who is, for example, an ovarian failure would have the potential to produce their own oocyte through this process using a stem cell?
Absolutely. And in a way, I'm glad you asked it. This is a point I clearly make when I speak about in vitro gametogenesis proper, which really was originally designed for the heterosexual couple. And yes, you can have individuals who, for one reason or another, let's say oophorectomy, you could, in principle, start with a skin cell, convert it into an oocyte, fertilize that, and transfer the embryo into a presumably pre-existing uterus, although the absence of the ovaries clearly will require hormone replacement therapy of one kind or another. There are probably other scenarios you can imagine, although I think more work will be required. For example, should we be thinking about the possible resolution of, say, a Turner syndrome? Well, you start off with an abnormal chromosomal complement. Can that be corrected in vitro? I don't know yet, of course, but I wouldn't rule that out. In a word, what today is being essentially bypassed with brute force, usually with donated material, could potentially in the future be refined and really be addressed in a more sophisticated manner. I would like to hope and pray that I'll be around for some or all of that, but knowing something about the calendar, I do have my doubts.
Thank you. It's all it's fascinating work. Thank you.
Thank you. I think it is important to every so often reflect about the future. We don't do that probably enough. We spend a whole lot of time on the present, which is important, of course, but here and there, a little bit of viewing the future is probably a healthy proposition.
Any other final questions, comments from the audience? No. Well, once again, thank you so much for being here on Zoom, if not in person, and for a wonderful lecture. We both lived through IVF. I guess it was, my memory is maybe it was 1968 was the first Nature article, and Louise Brown was in 1978, the first birth of an IVF baby. And then for so long, I remember, if it was an IVF baby born, it was like news, local news, to the point where it's now a common thing we practice every day. So one wonders, thinks that down the road, IBG may be something that's in the path, and maybe we'll be wrong, and it won't be 2 or 3 decades, but much earlier. Anyway, it was a wonderful talk, and very, very clear. And I think looking into the future is always fun and I think valuable too. So thanks again. Appreciate seeing you once more. And
my pleasure, Richard.
Bye-bye, Eli.
Thank you.